So maybe in a different world it might be possible for me to start this laboratory by bleeding some animal, maybe a rabbit or a horse. And if I were to take a vial of fresh blood, that is without any kind of anticoagulant added, and we're just to leave it in a test tube rack for even just a couple of minutes, what you'd see is that it would start to glop up. The cells would start to congeal, and you'd end up with this gloppy mess that consists of dark brown clots of blood, as well as serum. That's the clear liquid or clearish yellowish liquid that comes around the bits of clotty blood. Okay. And we've always known about this. I mean, people have been using blood for years to make things like blood sausages and morcilla. And we knew that if you don't keep stirring the blood or do something to keep it from clotting, you'd end up with this gloppy mess, which apparently isn't terribly good to make good morcilla. Now, if you ever had blood drawn, either because you were donating blood or because you're doing a blood draw for some type of diagnostic test, you might know that those little blood collection vials come with different colored caps. And the different colors on the top of the caps basically reflect the different kind of anticoagulant that's included. If you're just drawing blood for a blood donation, the anticoagulant that's used is sodium citrate. But there are other kinds of anticoagulants that might be used, EDTA or heparin or a couple of others that are commonly used. But if you collect the blood together with some type of anticoagulant, it doesn't turn into that gloppy mess. And you could take that tube and run it through a centrifuge, and you'd end up with a separation of cells. Right in the middle, you'd have just a little layer of kind of a grayish coat. It's called the buffy coat. And below that, you'll have the red blood cells primarily. And above that, you'll have a clear liquid that looks a whole lot like serum, but this is going to be different. It's plasma. And to make things perfectly consistent with your textbook, we would call that buffy coat together with the cells underneath it. We would call those the formed elements. And the formed elements would be including the white blood cells or the leukocytes, which are there in that thin grayish layer in between the plasma and the red blood cells. Now, to understand the difference between plasma and serum, we've got to take into account the composition of blood itself. From the lab manual that you've already read, you'll know that plasma consists of 3 different kinds of plasma proteins, together with water, of course. But basically, the 3 major plasma proteins are albumin, fibrinogens, and globulins. The albumins contribute to plasma osmolarity. Basically what it kind of does is modulate the effective saltiness of the blood without actually being a small solute like sodium or chloride ions. The fibrinogens are those proteins that are just ready to go to create a clot, basically kind of like protofibers that can stretch out and start to glop together cells and other things in order to form clots when clotting is necessary. And the globulins are largely those things that we usually call antibodies or immunoglobulins. And these are the ones that are relating to the immune system, and we'll be looking at the effects of globulins in our laboratory later on when we talk about ABO blood typing. The formed elements include the cells as well as platelets. Now, platelets, they're sometimes called thrombocytes. They're not really cells, they're kind of like fragments of cells that contribute to the formation of a clot. When you get those clots, it's basically the cells, the platelets, as well as the fibrinogens, which basically turn into fibrins. Okay? The cells include both white blood cells and red blood cells, leukocytes and erythrocytes, if you prefer. And in this blood sample that we've added an anticoagulant to and then centrifuged, the plasma and the white blood cells and platelets and the red blood cells kind of fall out in order. In the case of this blood sample for which we haven't added any kind of anticoagulant, the serum is basically the same as plasma, but it does not contain the fibrinogens, because the fibrinogens have turned into fibrin, and now the fibrins are going to be included in the clots. The fibrinogens that were there in the whole blood got turned into fibrins, which basically associated with the platelets in the cells to create these clots. And it's for this reason, basically, that serum is different from plasma. If you're doing a plasma transfusion, for example, in a hemophiliac patient, you might remember that's one of the X-linked conditions that causes a person's blood not to clot properly, we can ameliorate that situation by offering the patient a plasma transfusion, basically taking plasma with functional fibrinogens. That is, the parts that are kind of missing in the hemophiliac patient. And by adding functional plasma with fibrinogens, we're able to restore the normal clotting function that should be there in the patient's blood. In that sort of situation, serum just wouldn't work because it lacks those fibrinogens. Okay. Now, if you're interested in the globulins, however, if you're interested in collecting antibodies, the serum is just fine. Serum has everything that's there in the plasma, including the albumin and the globulins, but it just doesn't have the fibrinogens. So if you're actually just interested in collecting the antibodies, you don't need to bother with the anticoagulants. You could basically let the blood clot, pour off the serum, and use that serum as a source of immunoglobulins or antibodies. If you've ever heard of the snake bite antivenin, it's basically serum. Usually it's a horse that we've immunized against the snake bite venom, and we've basically just bled the horse. You don't have to kill the horse, of course. There's a lot of blood in a horse. You can just take off a little bit of blood, let it glop up, pour off the serum, and then use the serum as a source of antibodies against the snake bite venom that can be injected into a patient that's been bitten by a snake. Basically, the antibodies against the snake bite venom that the horse has can be used to help protect that poor guy that's been bitten by a snake from actually suffering the consequences of the snake bite venom. Okay, so in the old movies, it's the old doc and the cowboy flicks that say, bring me the serum because this guy's been bitten by a snake. And that's basically how they made that serum. Okay, so let's talk about ABO blood typing. Start off by drawing a red blood cell. Okay, so remember that the red blood cell is a biconcave disc. Basically, there's a little indentation in the middle where the nucleus was taken out. So it looks like a little bit of a donut with not a complete hole in the center. And it's filled with red stuff. It's packed with hemoglobin, the molecule that carries oxygen. Okay, that's a pretty good red blood cell, if I do say so myself. And outside of the red blood cell, we've got lots of different markers. We've got glycoproteins. In particular, there's a glycoprotein that goes all the way around the outside. It's called antigen H. And antigen H is going to be characterized by two different combinations of excess sugars. So if we look at one of these antigens up close, the antigen might look like this. It's got a little spot in which it's going to be attaching to sugars. It might have this sugar number 1. This is kind of like the A sugar. It's actually a monosaccharide. The monosaccharide is N-acetylgalactosamine. Let's call it sugar A. And if sugar A is there, that would make this whole thing basically an A-type antigen. Alternatively, the antigen H, might be attached by sugar B, which is actually just regular old galactose. We'll call it sugar B. Or the third possibility is for that glycoprotein to not have attached neither sugar A nor sugar B. And that's the combination that will give rise to the absence of sugar A or sugar B, which would be the O type. Now, the reason why the cell would have attached sugar A or sugar B or neither is based on the presence or absence of these two enzymes. One enzyme is going to be doing this. It's going to be attaching that sugar A to the polysaccharide. And that enzyme, call it enzyme 1. Whereas you might have another enzyme, enzyme 2, is going to have the effect of attaching galactose or sugar B to that polysaccharide on the outside. If you have neither enzyme 1 nor enzyme 2, then you're not going to be attaching any outside sugar at all to this little antigen H. And so no enzyme is going to be resulting in this outcome of having the absence of A or B. So basically, you might remember from our genetics discussion is that we had 3 alleles. We had IA, capital IA, we had capital IB, and we had little I. Those are the 3 alleles that we tossed around as we were talking about the ABO blood system. And it turns out that this gene here, capital IA, is going to have the gene product. When it gets transcribed and translated, the gene product is going to be enzyme 1. And so if you have this allele, you're going to be producing enzyme 1. The enzyme 1 is going to be there out there in the big world, and it's going to be turning at least some of these antigens on the red blood cells into A-type antigens. If you have this other allele, capital IB, then you will be undergoing translation and transcription. You'll be producing the second gene product. Basically, enzyme 1 and enzyme 2 are slight variations of the same enzyme, and part of the outcome of enzyme 2 will be to attach sugar B or galactose to the outside. So basically, if you had IA, IB, that's your genotype, then you're going to have both of these antigens on your red blood cells. This would be type AB. With this third allele, it's either going to be producing no enzyme at all, no functional enzyme, or it will be producing a functional enzyme that happens not to attach these sugars to the outside of this molecule. Either way, what you end up with is a polysaccharide that will carry neither the A nor the B specificities. Okay? And so here's where you might remember that you could have a big A, a little I. This would still result in type A blood because these glycoproteins are going to be carrying sugar A on them. Basically, we have the enzyme that would cause them to produce sugar A. Sugar B is going to be attached if we have at least one copy of capital IB. And the only way we can actually get type O blood, which is the absence of both sugar A and sugar B on the outsides of these glycoproteins, would be to have the genotype little I. And so now the question becomes, how do we tell the different types of blood apart? They've got these antigens on them, either the A or the B, or maybe both of them, like this cell, or they might have neither A nor B. How do we tell those blood cells apart? Well, it's going to be actually pretty easy. We'll be doing this in the lab. We've got the anti-A stuff. I think it's blue in color. We've got the anti-B stuff. It's yellow in color. And you mix it with the red blood cells, and they either agglutinate for a positive reaction or not for a negative reaction. But given that this is a biology class, you kind of need to know the background. How do we get there? This is kind of like the behind-the-scenes tour of an ABO blood typing experiment. So we've already talked about serum, and so you probably can guess how we actually came up with this stuff, with this anti-A stuff and anti-B stuff. There was an animal at one point. We injected it with the A-type antigen or the B-type antigen, and after a while it developed immunity, and then we collected the blood, and we let the blood clot up, and then we poured the serum, the liquid part, into another container, and that's the stuff that we're using to test our current red blood cells for the A and B antigen. So in that animal, after we injected it with the A or the B antigen, it started to produce an immune response. And the immune response resulted in the production of antibodies. And so the contents, what we have in these guys, are immunoglobulins against antigen A or immunoglobulins against antigen B. So if there's a reaction that indicates the presence of antigen A or B. And so now we have to think about, well, that immune response. Because after you're injected with something like antigen A, you start to develop antibodies against antigen A. And the wave of immunoglobulins that are produced, the first wave is going to be a population of antibodies that are called IgM. And IgM is a little bit different from your standard textbook antibody in the sense that it's got 5 parts. You already know that there's a Y-shaped structure in antibodies. Well, IgM is going to have 5 of these Y-shaped structures that are all kind of hooked together in a little ring. Kind of looks like a little ninja star. And remember that these points out here, these points at the very tips of the antibodies, the arms of the Y, are the antigen binding sites. And those are the parts that are going to be identifying and attaching to the antigens. And this pentamer structure, the fact that we've got 5 antibodies, 5 units, basically makes this molecule an excellent agglutinogen. It's able to come in and sweep up all the parts that are bearing the antigens relatively quickly. But it's also a structure that's relatively labile. It's not going to be sticking around in the bloodstream for very long. The second wave of antibodies that are typically produced are the IgGs. And IgG is your standard single unit antibody. And IgG has got a couple of advantages. It's going to stick around in the bloodstream for a lot longer, and in your typical immune response, we produce a whole lot more of these guys. And so IgG is going to be in there for the long run. IgMs are going to be there for the very first set of defensive sweeps, and IgG is going to be there second. There are other classes of antibodies as well. There are IgAs, which are 2 antibody units together, kind of hooked together at the base. as well as IgDs and IgEs that are kind of involved in other aspects. IgEs are the ones that we'll talk about later. They're associated with the sensitization of basophils, causing allergic reactions. IgAs are the ones that are associated with secretions like mother's milk, the passive immunity that comes as a result of a nursing baby. Those are going to be imparted entirely by IgAs. And remember that IgMs are the first ones, IgGs are the second ones. These are typically the antibodies that are produced in the circulating population of antibodies. Okay, so this is kind of important for us because the anti-A's and the anti-B's that we're using here, they are IgMs. In other words, they're particularly strong agglutinogens. They're going to be able to agglutinate things very, very quickly. So I want you to visualize this IgM, only a whole lot smaller. Now let's say this IgM is going to be specific for the B antigen. In other words, it's an anti-B IgM. I kind of remember drawing the B antigens in blue. So now what I'm going to do is I'm going to attach a whole bunch of these anti-B IgM antibodies to the red blood cell. And you can sort of see what's going to happen next. These antibodies are going to attach to the red blood cell, and they're going to make the red blood cell sticky to other red blood cells bearing the same antigens. So the effect is that all these red blood cells that might start off being separate, they're going to agglutinate, turn into these clumps of red blood cells that will all be clustered together. Kind of like a clotting thing, but the mechanism is really entirely different. So if you end up with these agglutinations, that basically tells you that the red blood cells are bearing the type B antigen. Okay. If we don't have any agglutination, in other words, if we have the antibodies and we get no agglutination, then we conclude that the blood cells don't have the type B antigen, so it would have to be either A or O. And so this is the underlying concept behind the ABO blood typing part of our experiment. So the next thing we need to consider is the RH factor. This is the plus or minus that comes after a typical blood type. Like O positive, that's my blood type, AB negative, A positive, etc. So what the RH factor is a different antigen. It's not related to the H antigen, which is marked with either A or B in the ABO blood typing. This is a different antigen, and we're going to be identifying it as antigen D, which is a protein that is there in the red blood cells. It's a different protein, so you can sort of visualize a different color protein coming off. This is the antigen D, and if it's present in the cell, then we would end up with a cell that looks like this. If it's not present, you simply wouldn't have those. Now, antigen D is a little bit different. I mean, for one thing, antigen D results in a different kind of specificity. Antigen D, for example, doesn't elicit a response that generates IgMs. We're going to be generating IgGs. And so the anti-D antibodies that we use in this experiment are going to be different from the IgMs in that they're not going to be nearly as good at agglutinating the red blood cells. Okay, remember, IgGs look like this. They only have 2 spots of specificity. And so the way this is going to work, if I make these guys smaller now, after these anti-D IgG antibodies are attaching to the red blood cell, they're also going to be sticky, but not nearly as sticky. It's not going to be nearly what we had before with all those ninja stars attached to the outside. It's going to take maybe a little bit longer time for the red blood cells to agglutinate with this weaker agglutinogen. And it's for this reason that, you'll notice it's going to take a little bit longer for you to develop the RH factor blood typing. Okay, so in this case, if you have the anti-D is there, present, you would say we are RH positive. If it's negative, if those D antigens are absent, in other words, you don't get any agglutination reaction at all, you would say that the cells are RH negative. There are a few interesting things that we can get into as well here. People talk a lot about the ABO blood typing scheme. You've got a lot of pseudoscience out there. For example, there's this thing called the blood type diet, which is based on absolutely no factual evidence whatsoever. There are some superstitions in some places like Japan, where they align particular personality types with type A blood or type B blood. At one point, a disgraced prime minister of the whole country stepped down, and as his excuse for his bad behavior, he said, "Well, I couldn't help it. I got type B blood." Right? That was hilarious to the scientific community. And at the same time, there's some really interesting results coming from these types of Now, right now we've got these things called genome-wide associations. Now that we have a lot of genomes sequenced, whole genome sequenced for a lot of humans, we can start to make these correlations between particular genes and diseases. And a lot of this was kind of led up to by some analyses like the ones I'm about to talk about. For example, if you look across the expanse, the spectrum of different blood types, A, B, AB, and O, there are some relatively well-established patterns. For example, if you have type A or B, basically either of these types of blood or type AB blood, there's a reduced risk of certain types of cancer. On the other hand, if you have type O blood, you've got a reduced risk of pancreatic cancer. Type O blood is also associated with a slightly higher vulnerability to the disease cholera, which is a bacterial disease of the gastrointestinal tract. One of the things we're trying to promote with BIO100 is the ability of students to distinguish between good, relatively evidence-based science, like what we were talking about with the genome-wide associations. You're going to hear a lot more about those in the coming years because we have such a wealth of information coming out. Every month there are like dozens to hundreds of new genes that are identified. And the press, the media kind of presents them as being, we've discovered another gene for a particular kind of cancer. And what's really meant by that is that we're noting an association. It's not a gene for cancer. It's not like you have this gene and you're going to develop cancer with 100% certainty. In fact, what it typically is that, yeah, you have this gene and you've got maybe a 8% higher likelihood of developing the cancer relative to people that don't have that gene. In almost all cases, there's never kind of like a sure ticket to a particular condition or the lack of condition. You could lack that gene and still develop the cancer. There's no guarantees. So as you're listening to these reports coming out in the next few years about, oh yeah, a new gene for this particular disease, a new gene for cholera resistance comes out, you might be actually talking about the gene that causes the A antigen, right? Because yeah, if type O blood really does have a higher risk of mortality or severe consequences from cholera, then that means that not having type O blood, in other words, having either the A allele or the B allele, is going to cause you to have a slight resistance to cholera. That would be another way of looking at the same result. So in this last part of the laboratory, we'll be looking at a right-stained blood smear. And we make this by taking a sample of blood from a patient and literally making like a one-cell layer thick smear over a glass slide. It takes a little practice before you can do this right. You have to get the thickness of the blood cells just right so you can spread out the cells and you can see individual cells. You don't want to have cells stacked upon other cells. And so this is probably something that some of you are going to do when you get to your physiology class. And then you let the smear dry and then you apply these stains. And one of the stains is going to be picked up by the eosinophils. That would be eosin. There's a basic stain. I'm not sure exactly which one they use. It's going to be picked up by the basophils. What we're going to try to do here is to look at a normal right stain to identify the different types of cells that are going to be there in blood. And there are nice pictures you can find if you just Google it up. But we're going to be looking for, well, red blood cells or erythrocytes, which are going to make up like 99% of all the blood cells that you see. These will be the little red dots. The other cells, little dots, are going to look different from each other. And there are 5 different kinds of leukocytes or white blood cells that you might be finding. And for the most part, these are the cells that we associate with the ability of blood to fight infections. Okay. And so the kinds of leukocytes we'll be seeing are neutrophils, monocytes, lymphocytes, eosinophils, and basophils. And the way we tell these different kinds of white blood cells apart, it's probably better for me to do this by actually showing you the pictures of the cells. We're going to do that in the laboratory. What I want you to have in the back of your mind is what each of these different kinds of cells are doing. For example, let's just start in the middle. The lymphocytes are the ones you've already learned about. These are kind of like the B cells and the T cells. B cells are the ones that are related to the production of antibodies with the adapted humoral immune response. And we've got the T cells But then we also have other kinds of cells, like the natural killer cells. There are other kinds of cells besides B cells and T cells, but for our purposes, we're just thinking about these two. We've learned about them in our discussions in lecture. The monocytes are the ones that are moving into and out of the bloodstream. These are kind of like the equivalent of what we call macrophages when they occur outside of the bloodstream. And they come into an area of infection and basically clean things up. They destroy extra bits of cellular dreck. That's kind of like their role. They'll also take some of those bits and present them to the B cells and T cells in the process of activating the adaptive immune response. Neutrophils are also there in abundance. These are also kind of like generic phagocytes. That basically means that they're eating things that are not supposed to be there. Neutrophils are there as probably the most abundant type of white blood cell that you'll find in a typical healthy patient's blood. And then we get to these relatively rare types of white blood cells. The eosinophils are the ones that will stain red because the eosin stain is picked up by the contents of the granules of secretions, the granules of potential secretions, the vesicles that are inside the eosinophils that are kind of waiting to be deployed against some type of invading agent. In the case of eosinophils, the target is some type of multicellular or a eukaryotic parasite. And you'll hear this frequently. You'll hear the idea that, well, one of the reasons why we get some types of diseases in human populations nowadays is that we live lives that are way too clean. We don't have exposure to parasites. And as a result of this lack of exposure, we end up having diseases like Crohn's disease and irritable bowel syndrome and various types of allergies that in the past were never terribly prevalent. The idea here is that before we had our eosinophils and other types of cells really active attacking things like parasites, but now that we live very clean lives, these eosinophils have nothing to do, so they start causing other kinds of problems. And then there's the basophils. These are the ones that are going to be staining purple. These are actually very, very rare. If you see any cells that have dark purple granules, let me know so we can share that with the rest of the class. Everybody in class gets to see basophils. They're rare. These are the cells that are going to be releasing histamine, which is the most important mediator of inflammation. So basophils are kind of like the equivalent, the circulating equivalent of those things that Sel Khan talked about in his video as mast cells, the ones that degranulate and release histamine, which cause the increased permeability of the capillaries in the area, allowing things like neutrophils and monophages and macrophages to come in and start doing their work and fighting the infection. Okay. We're not going to talk about these anymore. We'll be looking at them in the laboratory, but I do want you to know basically what they do.